Folks, welcome back. Now that we have spent all this time understanding microscopically what's happening with muscle contraction, hopefully you have a great grasp on that topic so we can proceed to back up and look at muscle physiology as the muscle is contracting as a whole. So the first part of muscle physiology I want to talk about has to do with muscle tone. And so let's talk about the length of the muscle. When a muscle is not acting, it has what we call a resting muscle length. And as we talked about before, a muscle can contract 30 to 40% usually in a normal contraction. And so the limitations, I know theoretically we said it could contract 70%, but we've got limitations put on the muscles because, like, remember, we're moving an insertion closer to the origin and we can only move it so close to it. So the bone movement is really what's limiting the muscle contraction. And so we're realistically looking at around a 30% of the resting muscle length to the contracting muscle length. Now, in addition, we know we can rest and overextend the muscle a certain percentage as well. Now, your brain is constantly monitoring this process and adjusting this resting muscle length, and we call that process your muscle tone. So when your arm is down, you can feel the muscle and you can tell whether or not it's relaxed or not. So what kind of receptors do you think are doing this adjusting? Yeah, keep that in the back of your mind. We're going to get to there shortly. So looking at muscle that's contracting, the amount of force behind each contraction is kind of dependent on that resting muscle length. And so we'll go into more detail for you to understand this concept. And it's called the length-tension relationship. There is an optimal length for giving the greatest amount of force during a contraction, and it's not a specific number. If you recall, look, I finally found something that I can use for a myosin. So it's the straw that bends. The entire length stays the same. Here we're in the cock position, and then when we have the power stroke, it's going to go to there. New ATP will bind, and I'll return back to the power position. So if this is the myosin and this is the actin, when the myosin is near the end of the actin and it contracts, it can pull, and you can get more and more overlap. But if it's at this end, there's very little more contracting that can happen because you're going to run out of actin. So if you're looking at just one myosin versus the actin, it looks like there's lots of movement that can happen. But remember, myosin doesn't act alone. There's a whole bunch of them turned at a 360. So I've just taken 4 now. And now if we put the 4 like this, and in this case, if the orange one was the red one, or in fact, I'll just add the red one and make it 5. If the red one is barely on the tip, when the red one contracts, that means, oh, actually that made it 6, I can't count. If the red one is the only one interacting with actin, that means the other 5 are not contributing any oomph to the contraction. So that is not an optimal contraction. You have to go until all of them can interact with actin in order to get an optimal contraction. And so here you would have optimal, and you would continue having optimal until the red one runs out. And even though there still is plenty of place for the blue one to contract, the red one has no one else to go. So you are limited to this much narrower range for normal optimal tension. So let's look at this graph, because that's what this graph is showing. So instead of starting on the left side of the graph, I want to start on the right side of the graph. The right side of the graph shows what happens when we are like this. Okay, if there's nothing, you can't make a cross bridge, then you can't contract. And that's what's on the far right side. And then we are starting to get more and more that can contract. And so that is what was happening in this area. This flat plateau at the top That's where everything can contract. And then what's happening on the left side is we have a very steep drop where nothing can contract because everything has reached its maximum. So the reason why it's more gradual on the far right is because you're going from 1 to 2 to 3 to 4 to 5 to 6, you're adding them up. And then at the end, you still have the majority contracting, and then it just plummets off. Okay. So I was showing this to you for an individual sarcomere, but you have to realize this applies to the entire muscle as a whole. So there is the area at the top, the plateau, that is called the optimal length. And in the optimal length, that means you have the optimal overlap. In other words, you have the maximum number of overlaps. You have formed the maximum number of cross bridges, giving you the maximum tension. Okay, so hopefully you got that concept now. Now, not only do you understand the optimal, you understand if we stretch it, force goes down, and if we contract it even more, force goes down. So this relates to muscle tone. Some motor units are always going to be active even when that muscle is at rest, because some are going to contract it to keep that resting tone normal. And this is an involuntary process, and I'll show you how that occurs really soon. Okay. So to understand this, go back to the concept of the motor unit, where basically we were talking shift work, and sometimes like motor unit A is working, and then maybe both B and C are working, and then maybe D is working. And so the motor unit's contracting causing enough to give it the resting muscle tone, but not enough to shorten the entire muscle. And so how we're getting the muscle tone is going to depend on what type of receptors do you think? Yeah, they're going to be proprioreceptors. And so there's two proprioreceptors we're going to be talking about. So refresh your memory, proprioreceptors are the ones that give you an idea where you are in space, okay? And so your muscles are kind of figuring out where they are. The first proprioreceptor is called a muscle spindle. And this is a proprioreceptor that's embedded within the skeletal muscle. And so if we look at it, what we have is we have a group of skeletal muscle cells, and they are ensheathed in a little bit of a realer connective tissue. And this acts as a functional unit where they're going to be monitoring the stretch of the muscle fibers in that muscle spindle. So are they stretch, are they contracted, or if they're stretching, is it a fast stretch or a slow stretch? And so we call the cells that are outside the muscle spindle, those are our normal things. They're called extrafusal because they are outside. Whereas the ones inside this muscle spindle, these are the ones being monitored. So these are the intrafusal fibers. So if you look at this picture, it's showing 2 green neurons and 2 yellow neurons, and that's just so that you realize that there are sensory neurons monitoring this, and as a result of what is integrated in the central nervous system, the green ones are the motor ones, and they are going to change what happens. So either you will have muscles fibers being stimulated to contract or to relax. So let's look at this. The pale pink, those are the intrafusal fibers. The blue is the sensory neurons surrounding them. When the muscle is relaxed, the sensory fibers are sending action potentials at a constant rate, letting you know it's relaxed. Everything is hunky-dory and nothing's going to change. However, when those muscle fibers are stretched, the rate of firing goes up. Okay, so that stretched muscle, okay, so that is an overextended muscle giving the message to the brain, hey, it has been stretched, we need to relax it. And in contrast to that, when the muscle fiber is contracted, it all becomes slack. So there's minimal, if any, firing going on, okay? And so that's how your brain interprets it, and it knows what to do. And so when we come back and we talk about reflexes, which is going to be, it's still going to be a few weeks from now. We're going to talk about reflexes when we talk about the nervous system, not about the muscular system. But this muscle spindle is going to be the sensory receptor that's going to be involved in that involuntary stretch reflex we'll be talking about later on. Okay, so that first proprioreceptor was the muscle spindle, which is in the belly of skeletal muscle, and it's actually monitoring the tension of a skeletal muscle cell. The second type of receptor that's monitoring is called the Golgi tendon organ. So yes, same Italian dude who identified the Golgi apparatus. And this proprioreceptor is not in skeletal muscle, it's in the tendon of the skeletal muscle. But it's not just anywhere among the tendon, it's in the tendon extremely close to where the tendon is next to the muscle itself. And so it's monitoring the tension within the tendon. So here's what it looks like. On the picture on the far left, you can see the Golgi tendon organ is very close to the skeletal muscle itself. And then there's a blown up picture in here. And in the blown up picture, you can see that the yellow sensory neuron is basically interspersed among the collagenous fibers, because this is tendon now, right? So it's dense, regular, connected tissue. Lots of collagen fibers all along one direction. And so the sensory receptors are interspersed among them. And so what happens is when the muscle contracts, that pulls on the tendon. And so if you have the neurons interspersed, they're all going to be squished because these collagen fibers are getting tense, okay? When the afferent neuron, the sensory neurons are squished, that's going to send a bunch of action potentials to the central nervous system, which is then going to have an efferent nerve, a motor neuron, which doesn't go to the tendon because the tendon can't cause muscle relaxation. So the sensory neuron is coming from the tendons, Golgi tendon organ, integrates inside the central nervous system, but the motor neuron goes back to the skeletal muscle associated with that tendon to cause it to relax. So both of these things, both the Golgi tendon organ and the muscle spindle can be monitoring at the same time, but the Golgi tendon organ is going to increase the Golgi tendon organ increases the impulses when the muscle is contracted, whereas the muscle spindle increases the impulses when the muscle is stretched, not contracted. So here is a slide that has both a Golgi tendon organ and a muscle spindle, and it's showing red, green, and yellow neurons. So make sure you understand what each of those neurons is. Pause it at this point and talk yourself through it. And with that, we are done learning all about muscle tone, and I'll see you shortly as we continue muscle physiology.